histogram, we're going to look at CD49F. This is our short-term hematopoietic stem cells, and this represents our long-term hematopoietic stem cells. From this, the CD45 RA negative, CD90 RA negative, this represents our multipotent progenitor cells. So this is how the hierarchy is identifying. If you want to identify a hematopoietic stem cell, you have to go through the properties of the side scatter, forward scatter. In this hierarchy, gate on Lin negative, 45 positive, 34 positive, CD38 negative, come down here, get the CD90 positive, 45 negative, and then off of CD49F, gate on it. Now, could you have done this in any other way? So the answer is yes. You don't necessarily have to do it in this order, but you do have to be consistent in whichever order you're actually going to do it in. The reason why we do it in this order is because what we gate on first are the things that are common between them, and then kind of split off from it. So this is common, so 45 and Lin is common, 34 is common to them, but then we use CD38 to distinguish between the committed progenitors and the more immature cell types, which are down here. So, if we just double click on the 34 positive, CD38 positive here, and then what we're going to do is we're going to look at CD90 and CD45RA. So the 90PE is 488, 575, 26. This one. Then the CD45RA ABC. This one is 633, 780, 60. Okay. So this is what we have. So this is CD90 along the bottom. This is CD45RA. Now, where do you draw the gates? Can you actually use the data that's there to draw the gates? So I would argue that you can as he uses, because you could argue that this is a negative cell population, this is a positive cell population, and similarly here you would have your cutoff so that this is a negative cell population and this is a positive cell population. So what you could actually do is you could say, okay, well, I could draw the gates. So this is my CD90 negative, CD45RA negative. So this is CD90 negative, CD45RA negative. And then over here, you have your CD90 positive, CD45RA negative, like that.
So how did you decide to cut it off at that point in like the 10 to 3?
You can do it based upon where this is.
Oh.
So if you based upon the scatter profile like this, you could say, okay, this is a negative population. You would think this may be positive up here, but because of where this positive cell population, you could say, okay, well, we could assign the cutoff here. Now, is that correct? Totally subjective. But what you should do is then go back to things like your unstained controls or your single stain controls. Go back and double check to see where you would put the cutoffs based upon all of your different controls. So you could go back and then you could reassign those gates. But what this is doing here is what is this population of cells here? So according to this gate here, these are the CD45 negative, Lin negative, here. Sorry, the CD45 positive, Lin negative. Then the CD34 positive, CD38 negatives. And then to here, this is CD90 along the bottom. So the CD90 on the bottom and the CD45 here, this represents our multipotent progenitor cells. So this is, according to the sheet here, this population represents our MPPs. So when we go to the data, let's go here. What this is showing us is that this is now starting to show us the number of cells that is present within each of those different gates. So we started off with this file, which had 6 million, but like I said, the actual number of cells that we were interested in was that off of that gate of the forward and side scatter gate, which is 5,835,603. When we get all the way down here to the MPPs, the total number of cells, which is your CD90 negative, CD45, RA negative, this only represents 5,074 different cells. So if we were to calculate what is the relative frequency of MPPs in this mononuclear cell fraction, what would it be? Well, the relative fraction would be the 5,074 divided by 5,835,603 times 100. What is that? Something quickly do on a calculator or a phone or something like that. Sorry? 0.08%. So, the frequency of your multipotent progenitor cells in the mononuclear cell fraction is incredibly small. If you were to then look at the percentage of stem cells, so we know the stem cells coming off the CD90 positive, CD45 RA, so it's coming off of here, there's only 344 cells. But then you're going to then have to subfractionate into the CD49F positive, CD49F negative. So the percentage of that is going to be probably another order of magnitude lower than that. So you're going to be looking at a percentage that's 0.005% or something like that of the total mononuclear cell fraction. So what we want to do is you want to go through all of these different subsets, placing in the gates, and then calculating the frequency of the 6 different populations of cells, which are then highlighted here. So the ones that are actually in blue being the MPPs, short-term, long-term, and then off of the CD38 positive, CD34 positive, 38 positive, you want to then get off of here and do this gating strategy to identify CLPs, GMPs, and CNPs. What we want to see in the assignment is a kind of gating strategy laid out something like this, and then we want you to calculate the frequency of these cell types in the cord blood mononuclear cells. So that what you're getting is you're getting this number here, or whatever it is, as a fraction of the mononuclear cell count, not of the total number of events, but off of this one, and then you can actually get, and you can then identify what is the relative frequency of the hematopoietic stem and progenitor cell populations. In this case, it's actually available cord blood, and is there any variation between the 4 different cell samples? Yeah.
So do you want us to set the gates for one sample and then apply the same gating strategy to all of them, or do you want us to go through and gate each sample separately?
Yeah, so so the answer is, in generally, yes, you would apply the same gating strategy to all the different samples. Now, the reality of the situation is that because these are biologically variable things, they came from different people, you might actually see slight differences in the gates or where you should place the gates, just simply because maybe there is a little bit of variability between different samples. So you need to go back, you need to check that nothing looks, totally weird. But yes, in general, you always want to apply the same gating strategy. You don't just want to keep on moving the gates to make it look awesome. So does that make sense? Kind of. Not really.
For this, so I understand this frequency, but then you said at the end we're supposed to calculate a relative frequency for everything?
Yeah, so if you look at the panel that you have here, I just did it for the MPPs.
Yeah.
What you also have to do is there's a gating strategy that exists for the long term, the short term, as well as this. So for those 6 different populations of cells that are indicated here, you need to find out the relative frequencies of each of those 6 populations of cells.
Okay. So 6 relative frequencies.
Yeah, but
They're
everything will
okay.
relate back to they'll all have like the same common denominator in a way. They'll all have the same number for this, but you'll just get a different, population. You'll just get a different number according to this.
Okay.
There is the best way, or the best thing to do is for you guys to just go and try it, see what happens. Like I say, there's also the class again tomorrow. I think it's from 10 to 12. Please come along to that. If you have any specific questions or anything as well, or you just want to go through it again, yeah, come along to that and ask any more questions. And then I think most of you, well, my office is on the 4th floor here. Again, my office, I'm more than happy for anybody to come and ask any questions. So please do that as well. Tomorrow I'll probably be leaving early-ish at about 3:30 because my kids have their school sports day. It's like their inter-city track meet or whatever. So I'll probably be leaving around 3:30 tomorrow, but apart from that, if I'm not here in class, then more than welcome to just come up, stop by, and we'll go over it. So this is kind of like, the reason why we're giving you this is because this is using flow cytometry in the, I don't want to start like in the real world, but this is the actual data that we collected from cord blood, where what we were trying to do is we were trying to identify what is the relative proportion of the different subsets of cells between different collections of cord blood. So that is what this is. This is what this data is. It's taken from actual data from actual cell samples, where the postdoc in my lab, Xiaoying, she actually went through this and did exactly this on all of the data. So it's not just we're doing it just to make an exercise up for you guys. This is something that we actually did and tried to identify it, and then you will hopefully be able to see the variability that exists and the differences that you see between different collections of cells, and try to identify the frequencies of stem cells in it. Now, if you can do this for the hematopoietic stem cells, you can do it for any other stem cell system that you can think of, because it is the hematopoietic stem cell system that has one of the most stringent immunophenotypic characterization of those cells, just in terms of the sheer number of different parameters that we need to use to identify those cells. Most other cell types are like 2 parameters. Oh, we're going to use CD49 and we're going to use CD44. Here we have multiple parameters that we use to identify the cells. So we're giving you the hardest one to do, but you can do it. You can do it for anything. Yeah.
I just have a question about the compensation
Oh.
thing. Is there a way to check if I did it right, or because you said it was
The best way to check